Okay. Yo, let's get started, guys. How are you guys? Good? So that guy that you saw here is Brandon. He's the brother of the CSO. He's the CEO of the company. Pretty young CEO. He was just chilling here because he's a friend from long time ago. Okay. What did I want to say? So I'll be posting the solutions to the problems today. I just didn't have time because I had some work to do for research to submit this morning. And then what else? Did you have any issues with the quiz tutorial? No? Schedule? Everything's good? Yeah? Next tutorial. So I'm gonna, yep. The TA. Yeah, the TAs will grade it. We talk and we're gonna build probably the solutions to it. Well, not the multiple choice, but the short answer question together. So the short answer, I'm asking for 3 things. If you got the 3 things, makes sense, right? Easy exam stuff. But did I want to say? I'm gonna answer also a question that you're probably gonna ask me in the next few minutes. So next quiz will be on the topics you covered in this week's tutorial. Yeah? I have to review it. Yeah. I don't even, so the questions, I'm just reusing stuff, so I don't even remember what that question is related to, but I will check it out. So if you've seen the first quiz, right, were those questions somewhat simple? It should be along those lines. I'm not going to ask you like, super long questions during a quiz, okay? But yeah, I don't know. The TAs will be giving you your grade. The TAs will be also giving you the quizzes. You don't get to keep them, so take a picture of them and give them back. The reason for that is that we have to keep all copies of your exams, quizzes for I don't know how many years. Some profs don't do it, I guess, but I'll do it just in case. Okay? Sorry? Yeah. It's just I need to keep the copies, physical copies, in the office for, I don't know, 5 years or something like this. Same thing as your taxes and all that, okay? What else? Midterm in 3 weeks. It's getting close. Are you guys feeling scared or you're getting everything? Yeah? A little bit. Yeah, not too much, not too much. I'm not crazy on killing people with questions, okay? The goal here is to understand and get a good grade and then apply to med school, okay? Not not fail and then end up like me, doing a postdoc. I swear, man, it's been like 10, 12 years of university. I'm getting tired of this, but soon, we're gonna have our own lab, okay? So what did I want to say? So today we're gonna review a little bit the stuff from last week. The last lecture, specifically the 3-point test cross, just to make sure that you guys understand it properly. Then I'll ask you some questions about it, and then we'll get into statistics. So chi-square test. Hopefully you guys are awake for that, okay? Okay, so remember this here? Right, so when we're doing genetic mapping, okay, I'm gonna review these things here. When we do genetic mapping, okay, we are trying to map 2 to 3 genes between each other, not more than that, okay? When we're using the old method, okay? So your goal is to try to find the distance between 2 genes, sometimes 4, but very rarely, because you'll end up reaching that 50 centimorgan, and it doesn't work anymore, okay? So when Mendel did his, not Mendel, but what's his name? Morgan did his experiments, he found that the recombination frequency is it's proportional to the distance between 2 genes. So 1% recombination frequency equals 1 mapping unit or 1 centimorgan, okay? If the genes are linked, the maximum you can have is 50 centimorgans, okay? Other than that, then it becomes treated as independent assortment, okay? So if we, actually it's Sturtevant, the student of Morgan, that found that. So if you look at 3 genes, for example, A and B and A and C, and then you see that there's 5 mapping units between A and B and A and C, you can deduce that the map looks either like this or like this, right? And so for you to find that, you need to look at the double recombinants, which will show you where the double crossover happens, okay? So last week I showed you a table like this. Last lecture, man, I mean last week. Last lecture I showed you a table like this, okay? And in this table, even if I don't tell you what the crosses are here, just with this table you're supposed to be able to figure out which ones are the parental lines, which one are the double recombinants, and which one are the single recombinants, right? And the way you do this, and it's logic, right? Because if 2 genes are linked, there's more chance that the 2 alleles will go together than there's a recombination happening between them. So a parental line without any crossover is more frequent than a single recombinant, which is more frequent than a double recombinant, right? So in here, if you're looking here, these are the parental lines because you have 580 gametes that are of this genotype and 592 of this genotype. Automatically, you can find the double recombinant, which are these ones here, the smallest of these 8. And then the rest are the single recombinants, okay? First thing that you do when you get to this point, you will probably have a table like this or something missing in there, and then you'll fill it out and then end up just choosing an answer from the list of answers, okay? The multiple choice. So what you have to do is take them 2 genes at a time to be able to build this map. So the first thing that you need to do is identify the parents, double recombinant, single recombinant. The gene order here does not mean anything, okay? This does not mean that CV is in the middle, okay? It just means that CV plus and CT plus. So here you have to think in terms of genotype, not in terms of sequence of genes, okay? So to find the gene in the middle, you ignore everything except the parental and the double recombinants, okay? Because the double recombinants, there's 2 crossovers happening. And because there's 2 crossovers happening, only the gene in the middle will change. The other 2 will stay the same because it will go like this, crossover, so there's a change in the middle one, and then cross back over up there, okay? So if we look at the parental, we have V, CV plus, CT plus. And then if we look at this double recombinant, we see V, CV plus, so these are the same, and then the CT changes. So automatically you verify that with the second one, V plus, CV, CV, CT, CT plus. So you know that CT is in the middle. So that's the first thing. Now what you're going to do is compare your 2 other genes to this gene in the middle, okay? So you're going to look at V and CT and CV and CT, okay? To find the distance between this pair of genes and this pair of genes. So this is just what I just said here. So you have multiple possibilities of a gene in the middle, either the CT gene, the V gene or the CV gene. And then if you do your double recombinant, you'll realize that these ones are the same genotype as the double recombinant, okay? So that's that. So the top one is the only one that's plausible. Now let's find the distances. Step number 3, right? You find the distances. So start by one of the 2. So let's say, for example, V and CT. Forget about the rest, and forget about CV in this line, in this side here, okay? So you look at the first one. The parental is V, CT plus, right? So, you'll have it like this, okay? So you look at these single recombinants, and you find the one that's different from the parental. Okay, so V, CT plus, it seems like this one is the same, right? So it's not recombinant for V and CT. This one is the same as the other parent, so it's not recombinant. And then if you look at the other 4 here, they're all recombinant for V and CT, okay? So to find the recombinance frequency, you add all those that are recombinant for V and CT and divide it by the total gametic progeny, and you multiply that by 100 to find a percentage. This percentage is also equivalent to 13.2 centimorgans or 13.2 mapping units. Does that make sense? Good. Why is the maximum recombination 50%? Well, let's say, for example, you had two classes that are 250, 250, and then you divide that. So they're exactly the same, right? You'll find that the parental is also 250, 250, so that gives you a total of 1,000, so it's half of that, right? If you do it like all equal and not linked, you'll find that 50 centimorgan is the limit for independent assortment because they have all the same number of gametes between them, so 250, 250, whatever, okay? Equal proportions. So we found 13.2. Then you would do the same thing, right? You would do the same thing for CV and CT. So you find the ones that are recombinant, you add them, divide by the total, multiply by 100, gives you 6.4. So that's 6.4 centimorgan. So now we have, yep.
Can you review the difference between centimorgan and a mapping unit?
Same thing. Just two ways of saying different things. Same thing. Two different ways of saying the same thing. What else? So now you have 6.4 for these two loci, or loci is just a place where locus is just a place where a gene is found. Okay, loci is a plural of that, so two genes. So now you have your map like this because you found the middle one, you found the two extremes. Now to find the distance between V and CV, there's two ways to do it. Either you add 6.4 to 13.2, and that gives you 19.6, right? Or you go to this table and you start counting, doing the same thing, finding the recombinants. The problem with that is that the double recombinant, it's hard to see. You don't actually see it because the double recombinant looks like the parental, okay? So don't do it by counting these because even that, it will give you a wrong number here, okay? This is what I was trying to explain last time. So don't, for the extreme genes, don't use the numbers here, just add the two distances, okay? Is that clear? Questions? Okay. No questions, let's test you. Let's say I give you this on the exam now, okay? And I present the data in this messed up order like this. Which ones are the parental? Yeah? 302 or 308. Why? Most frequent, right? Okay. Now, which ones are the double recombinant? 10 and 11. Look at that. We have colors today, okay? So, these ones are the parental. I just put this like this. Took it from another prof, so it's easier for you guys to see, okay? And then the double DCO, double crossover, but same thing as double recombinant, okay? So, are the smallest ones. And then the rest are single crossovers. If you look at the single crossovers, 82 and 77 are closer, right? And then 101 and 109. So these two are probably for the same gene, and this one with the 77 are for the same pair of genes, okay? Which one has the biggest distance? These two or these two? Sorry? B and C and B are further apart? A and B are further apart from each other. So let's see the parental, A and B. Yeah, because A and B looks like this or like this. And if you look at these two, it's a crossover between A and B because the genotype changes. So yeah, you are right. A and B are further apart. Did you do this before? Damn, that was good. Okay, cool. So what you can do is fix it like this or just do it like this. I don't think it's going to be too complicated, but if you do the calculations, I'm a little bit sick, sorry for that. And if you do the calculations, you'll end up finding the whole population, and then probably another 1,000 here. Yeah, this is good. Okay. So if you do the calculations, you'll find these distances. Make sense? So then, by finding these distances, you can also just find the gene in the middle, right? Able to do that from here? Yep.
I didn't get how did you choose the farthest ones for the calculation.
Okay. So if we look here, we have the parental ACB, right? So ACB is 2 dominant alleles, right? So then if we compare, let's say this line here, the B changes, right? So this is a crossover between the gene A and B. Does that make sense? Wait, what are we doing here? Wait, let me just read this, maybe there's a mistake here. So ACB, sorry, this one goes with, so the 308, if you compare the A and B, that's where the A changes, right? Okay, and then if you look at this one here, you should be comparing it with this one, right? So, wait 1 second. So A and B, and then you have A and B. Sorry? Yeah, let's do it, guys. Let's do it. Okay, we're looking at A and B, right? So let's remove all the C's. Okay, so these are the parental, right? So which ones deviate from this? This one looks the same, right? So this one clearly did not have a crossover with this one. Let's look at the other parent, this guy here. Clearly this one did not have a crossover with this parent. So so we can remove this, right? We can hide this, and we can hide this. So what's left? We have 101 and 109, right? So if we look at this genotype, this one is different from it, and this one is different from it. And if we look at this one, same thing, right? So these are the crossover. These ones are the crossover between A and B. Is that clear? Good. So these numbers, you're gonna add all of these to find, and then divide by the population times 100 to find the distance. Okay. And then you can do the same for the other one. I'm not gonna do it now, but it's a bit annoying because when you have it like this, like you have to kind of do it mentally, but in the exam, maybe just put your hand over like 1 letter, do 2 letters, and then play with it, okay? It's gonna be easier for you. Does that answer? Yeah. Okay. All right. So now a little bit of math, okay? Sometimes I can just give you the distance between the genes, and I ask you what is the probability that you'll have a double crossover, or what's the probability, or how many of the offsprings, the progeny, the gametes will be a double crossover, right? So if I give you the distances, to find the probability of a double crossover, you need to find the probability of a crossover between A and B times the probability of a crossover between B and C. Does that make sense? Because you need the 2 events to happen at the same time, okay? Remember, this is can be converted to a percentage, right? 23.1 centimorgan is the same thing as 23.1% recombination, right? And 23.1%, if you put it in decimal, it's 0.231. Basic math, right? Okay, so what you do is you take these probabilities, if you put them in decimal or in percentage, it doesn't matter, and then multiply them and you'll get this number here, or 4.1% chance that the population will have double crossover. So let's say if I give you a population of 1,000, then you're expecting to have 41 of them as a double crossover. Does that make sense? Simple? Yes. Good. Thank you for answering. So in this case, we observe only 21, right? Because we have this one here, 11 plus 10. So there's something that's deviating from the expected number, okay? When we see something like this, we can say that there's interference between the crossovers. So there's one crossover preventing the other, or not preventing it, but making it harder to cross over a second time, okay? Sorry for that. And this just comes from the fact that, So the chiasmatas, when they form, it's something physical. It looks like this, right? And so it would be hard to imagine having a crossover right here, right? So the bigger the distance, the more chance that you will have a crossover, the closer, the less chance, okay? So this is just some structural stuff. Because crossovers happen like this, it's very hard to get one closer to the other. You can have interference between the crossovers. And that is why the double recombinant is less than the single, which is less than the parental. Okay? Yeah? Well, it's useless. Depends what you're doing here. But you know, if you're studying crossovers, I guess, yeah, it's more of a statistics thing than anything else, right? It's more just to make sure that your numbers make sense, are happening because of something and not just chance or whatever. Okay. Let me see where it could be useful to calculate interference. So, if you know that you're studying two genes that are very close to each other and you are trying to figure out if there's a possibility that both could be messed up because of a crossover, you can kind of do that statistically by figuring out if one crossover will affect the other or not, right? I don't think researchers really use this. This is like prehistory Mendel time stuff. Well, not Mendel. It's close to now. It's like maybe 70, 80 years. But now we don't even see this stuff. So I just look at sequences and peaks, and I have no idea what the hell is happening like in the cell, right? Because of computers and all that. Yeah.
Can you explain, please, why we chose A and B and B and C?
Why did we choose A and B and C?
Yeah, when we did the slide right after.
This here?
No, the one right after.
Where?
Yeah. What's the reason behind choosing A and B?
Okay. So first, the gene in the middle is important in this case, right? So if you have the gene in the middle, then you have the two regions side to side, right? So it's the probability that there's a crossover happening here, and the probability that a crossover happening here, and you multiply them, right? So from this, you need to find the gene in the middle first, right? So you see this one here is 41.1. So you can see that these two are the extremes. And then the two in the middle are A and B. So you would have a B here, and then B and C. So you'd have, B in the middle. Yeah. Thank you. B in the middle, and then, yeah, so this one, A, B, C, and then you have AC, which are the extremes. Yeah, exactly. Sorry for that. Thank you. I'm learning them as we go. Where did I want to say? Okay. We can calculate interference. Okay? And the way we calculate interference is with this simple formula. So if we do, so interference is equal to 1 minus the observed over the expected. Okay? Observed was 21, expected 41. If you, and that we just saw that, right? So then if you do that calculation, you end up with 0.49. If you had the same amount as observed and expected, the interference would be 0, right? If you have 0 here, and then, I don't know, any number here, then you'll end up with interference equals to 1, right? So this is the maximum interference that can happen. We can say that the other one is blocking, one crossover is blocking the other one, okay? Yeah.
What does interference represent?
It's just arbitrary units. It's a way of saying, like, are we closer to 0 or to 1? And then it's telling you if there is kind of some sort of inhibition between one crossover and the other, but it does not have any unit. If it's close to 100 to 1, you can think of it as there's 100% interference. If it's close to 0, then it's 0% interference, and so 2 crossovers will happen. Okay? Yep.
Is it possible to observe
More than expected. That's a very good point. So How would you count gametes in a negative way? There's no way, right? No. So no way to have a negative number.
So it should always be like a less or equal or less.
Exact. Equal to 1 or less. Because you're observing you're observing gametes, right? So if the gamete is there, you're observing it. If there's no gamete, 0. There's no way for you to have minus observed or minus expected. Yeah?
But I think the thing is like if there's more observed double clusters than what we expected, then
Oh, I see what you mean. So if there's more observed,
Propagation cluster would be like a negative.
very good point. So if there's more crossovers than expected, you'd end up with a negative interference. I think it's just not possible, like physically, so they don't account for it in the equation. But
In that case, the interference is going to be positive level crossovers, not the opposite way.
yeah, exactly. So it's like the opposite of the concept. But I don't think you can observe more double crossovers than expected. Yeah, I don't know. That's a very good point. Never thought of it this way. So,
I'm not sure.
Yeah.
I'm just thinking,
Yeah.
probably would assume like they're linked together or like that they go together. Or
if there's double crossover,
like if there are more observed
yeah,
than expected, if a negative number, then you would assume that probably both the genes are
more linked than before. Yeah, no, I can't even imagine it mentally, right? I don't, just assume that there is no observed more than expected. Okay. Actually, I'm going to Google this after, see why. Because then I've 12 years of dealing with this, and I've never thought about it in the negative sense because it never happened in front of me. So, but yeah, is anybody in math here? We can figure it out. Okay, so if you have multiple genes and you're trying to, this is a question that Joe asked last time. If the max is 50 centimorgan, how do you end up mapping the whole thing? Well, you have to treat it like separately, right? So you would have multiple maps and then join them together, right? And you can draw the whole chromosome like this by starting, let's say, for example, these two. So 40, no, you can't do these two. Oh, you'd have to do them separately and then put them all together. 2 by 2, 3 by 3, depending on if it's more than 50 centimorgan or less.
If you have 4, how can you figure out which one is in the middle?
If you have 4, you'd have to separate them as 3, 3. And then you'll find the one. Huh? Yeah. Yeah. Yeah.
usually the question is you find the distance. So
Yeah.
so you find them between and add them up, and then they would give us
No. I'm not talking about questions. This is just the concept of the maximum recombination frequency or distance is 50. That's it. So if you want to map multiple genes, you need to do multiple maps and then put them all together. Make sense? Like usually by 3 because you need a gene in the middle and then you keep crawling, right? But no, I would not ask you something like this. What I could ask you though, and we're going to try it, I think it's the last slide. It's like giving you distances and then you have to find the gene in the middle. Okay? Yeah. So if the distance between two genes is 50 centimorgans, that means there's equal numbers between the parental and the recombinant because there's 50% recombinant, 50% parent. This is independent assortment, right? So that's the limit for studying linked genes. After 50%, it becomes unlinked, okay? So when you have, as a scientist, to draw the whole chromosome, there's thousands of genes, right? So for sure it's more than 50 centimorgans. So the way you do it is you take two genes that are close, measure that, find a third gene linked with the other one, measure that, and then do it step by step. That's what I mean. right? And this way you can figure out the length, which is 79. But I think these numbers don't add to 50, right? Yeah, they don't add to 50. So this is wrong, but, you know, yeah. CF9 and BK. Let me just correct this because it doesn't make sense. 18 plus, yeah, so this is wrong, right? Yeah, I understand. But for you to be able to do it this way, you need to find the, because this one, if you add it to this one, it's more, right? So you see what I mean? You have to find ones, yeah, exactly. It shouldn't be more. So let's just do this one here, 10. And then, I don't know. CF9 to HLBK? Yeah, that works. You could do it separately like that, yeah, true. Yeah, true. So if you take them 2 by 2, then this thing works here. But if you take them 3 by 3, this doesn't work, okay? So you can take basically CF9 and HL, okay? And then HL and BK alone, and then BK and RD22 alone, and then find this, okay? In this sense, it works. But if you're going 3 by 3 genes, it doesn't work, okay? So if I just, yeah, that's it.
So if you take them one by one,
It works.
18 is less than 50, it's okay.
Yeah, exactly. Because you know that CF9 and HL are 18 units apart. So then you forget about CF9, you look at the distance between HL and BK, that will give you 37. So you know that CF9 and BK, the distance between them is 55. Okay? Yeah? Well, there's only 2 ways to go, right? You can either go this way or this way, but you'll always have one gene that is common between the 2, right? So in this case, if we're looking CF9 and HL, and then we have a different distance between HL and BK, because you can technically just do those numbers that we did earlier, just for HL and BK, and you'll find the distance because you'll have recombinant gametes, you'll count, and then you'll end up with this. That's a dihybrid test cross instead of a trihybrid test cross, okay? And so you'll have the distance between HL and BK alone. Then you can do the same experiment for CF9 and HL alone. You count the gametes, and then now it's just a question of is BK on this side or that side, but that's another story, okay? Make sense? Questions? I saw some hands up, and then they disappeared. Yes, Joe? Yeah, we did in the beginning of last lecture. It's the same thing, same exact thing. Exactly. So you just don't find the gene in the middle, but you can still find the frequency, recombination frequency, right? And that will tell you the distance. I think I can go and bring it up. I just think it was here. Time is it? Okay. So we did this, right? So then if you count these recombinants, add them, because there's 2 genes, divided by the total, multiply it by 100, it will give you a percentage, which is your centimorgans. This one is easy. It's when there's 3 genes and 4 that it starts to become annoying. Right? No. I'm not mean. We'll do a 5. No, I'm kidding. Okay. Stats. Let's do some stats. Scientists love stats, okay? Because, it's something that you can, in your head, imagine, right? Probabilities and all that. If you're going to a poker game and you see that the guy has a royal flush 3 times in a row, like, brother, you're cheating, you know? But scientists need numbers on that, so they use statistics, okay? My most hated topic, but it's okay. So if I roll the dice 10 times, okay, in a row, so this is just a little bit introduction, and then we'll get to the point. Obviously, if I get all of them ones, clearly I'm cheating, okay? Like, you don't need to do the chi-square test to figure this out, okay? But sometimes it's not as clear. Sometimes it's not as clear, and it's not clear when you're doing trihybrid cross because sometimes the numbers could look like they're equal, but they're not. So for that, you need the math, okay? So to figure out how lucky you need to be to get 10 ones in a row, you have to do the probability that you get 1 times probability you get 1 10 times. And so that's 1 to the 6 times exponent 11, which gives you 1 chance in 60 million. So you are clearly cheating, okay? Obviously, there's a chance, right, of it happening, but, you know, the more plausible answer or more plausible cause is the fact that you are cheating or something is happening. Okay? So let's do some genetics now. So let's say we want to see if the what's the expected chance that these genes are linked or not. Now it's a bit more complex, right? Because now we are not sure if these are equal proportions or not, and they're very close numbers, right? So it's a bit hard to say, but even then with the eye you can say that. You can see it, okay? This is for very small numbers that are easy to process, but for example, in research, the numbers are super huge, okay? And it's complicated to do just by using your eyes, okay? And usually it's not forced like this. It's a whole Excel sheet of like thousands of lines, okay? That looks. Where is research? So I'll open this while it's loading. Yeah, it's going to take time because you've seen Excel, but I'm showing you a little bit of research stuff, okay? Just by the speed at which it's loading, you should like imagine in your head that you can't do it by your head, right? So there's no way, okay? No way to figure out if this is linked, not linked, what's happening, okay? So this is why we do statistics, not because you want to see if you're cheating at dice or not, okay? So we use something called the chi-square test to test this. I skipped it in chapter 3 because then we're going to talk about it again in chapter 4, so I combined both of them together. This way we don't waste too much time on the math. But this is the equation. Did you guys read the book? Anybody? No? Okay. So I'm going to explain full-blown explanation then. Okay? What the hell? Why is it zooming? Okay. So the chi-square equation looks like this. Okay, so you have to, again, we're talking about the observed and expected in the same principle that we just did. In an independent assortment problem, you're expecting the parental and the recombinant to be exactly 25% each for a 2-gene, right? So in here, because we're doing a 2-gene thing, if it was independently assorted, you would have 250, 250, 250, 250. And then the total, which is 1,000, okay? In that case, not this stuff here, right? If it's 3 genes, you'd have 8 different categories, and then they all need to be equal because we're talking about independent assortment. When you're talking about linked genes, those numbers change. You have 2 parental that are high frequency, 2 double recombinant, etc. But sometimes it's not clear because the numbers look alike. For example, this one here, okay? You're not sure if it's linked or not linked. So what do you need to do to figure this out? You need to do the chi-square test, okay? So the expected, so you're going to go over each category, okay? The expected for this one is 250, but we observed 325. So here it's 325 minus 250 squared divided by 250, okay? And then you add all of them. So you will have 4 times this thing here, okay? Which is right here. So initially the idea is to figure out if they are linked or not. So you need to make a hypothesis. I like to call it the null hypothesis, but useless words. It's just a hypothesis, okay? G and T are not linked. So I think they're not linked, but you guys probably do think they are linked, right? Because they look very close to each other. So then you calculate the expected, sorry, not 250, 300. You calculate the expected value for each one of them. Because we have 1,200 total, each category should have 300 if it's independent. Is this clear up to now? Simple? Yeah? the only one you can do is unlinked, right? Because that's the only one you know the proportions of it. The other one you cannot really have the proportions for it. It changes. But the linked is clear, 25, 25, 25, 25, okay? And then you have your expected values, so then you calculate the chi-squared thing. So usually what you do is you build a table like this, and then you just fill out the table. I don't know how I'm going to do it in the exam because it's multiple choice, so maybe it's going to be a bit different. We'll see. I'll figure it out. But you're going to do your observed, so that's just the table that we had earlier. The expected 300, and then your calculation O minus E gives you this, squared, and then divided by E, right? Gives you this, and then you add them all up for all the categories. That gives you a 12.26 chi-square value. Now, so we have these expected values in chi-square, okay? We use these expected values to see if something is by chance or not, if it supports the hypothesis or not, okay? So we have this number that we just counted. Now, for us to compare our data to the expected values, we need to figure out something called the degree of freedom. And this is just a number that you need to figure out. And the way you do it is by finding the number of classes that you have minus 1, okay? So here we have 4 classes of gametes. So 4 minus 1 gives you 3 degrees of freedom. It's just an arbitrary number that helps you when you're looking at the table of expected values. For example, if I'm throwing a dice 6 times, my degree of freedom is 5. Like a dice that has 6 sides, my degree of freedom is 5 because there's 6 possibilities, and then minus 1 gives you 5. If I'm playing rock, paper, scissors, my degree of freedom is 2 because there's 3 possibilities, I remove one of them, okay? Why do they remove something, one of them from them? There's an equation that you have to do to figure out. If you take biostats 322, I think, you're going to learn that. Especially those that are going into research, it's important to take biostats, okay? Yeah.
At the beginning, you said why do we use it, the chi-square, not the sum?
In the sense, so if I tell you, like, I get 10 times a 1 while playing dice, it's clear that it's yeah, I'm getting there. So in BIOL, it's a bit more complex because this here is not clear if it's independent or not, okay? And if they're linked or not. But it's used for many other things. We'll see in chapter 14, I think, or something, when we talk about population genetics. It's used for other stuff, okay? It's just to see if something is happening because it's expected to happen like that, or it's happening because there's something affecting it, right? You can even count interference using this, okay? When you're expecting and not expecting, okay? Which we did earlier. That's pretty much it. It's just to take data that does not look clear and then determine something from it. That something depends on you. What do you want to do? As long as you have you're able to figure out the expected numbers, then you can study pretty much whatever you want, okay? You can even use it to figure out which gene is in the middle, but that's your hypothesis. I think it's this one, then you go put the numbers there, you'll figure out if it's in the middle without doing the other way that I showed you, okay? It's just we don't need to get into this because, too much math, okay? Usually, why am I gonna use this chi-square? Well, obviously for what I'm teaching you, right? Are they linked or not? And then you figure it out, okay? I'm gonna give you some numbers that look like this, which you have no idea if they're linked or not, and then ask you to figure it out, so you need to do exactly this. The table there, the percentage of accuracy? Yeah. Getting there. Right there. Yeah. Linked and unlinked. So if you're trying to figure out if the genes are linked, then you have to do your null hypothesis is they are not linked, and then the expected values for each category is 1/4 of whatever population you have, right? And then depending on what you find at the end, you can then determine if it's linked or not, okay? So to figure out if your data supports your hypothesis, which was, I forgot, which was G and T are not linked, you're going to use this table here, which I will provide you probably in the exam, which is in your book, I think page 90 something, 97 or 87 or something like that, okay? So we have 3 degrees of freedom, right? Because we have 4 different categories, -1 gives you 3, so that's degree of freedom number 3. And then we found our chi-square to be 12.26. So if I look at 3 and go up to here, we reach here, right? So the chi-square that we found is bigger than this number, okay? Which means that there's 1% possibility that this is happening by chance. So let me re-explain that because I don't want to mix you up, okay? So how do I explain this? Just give me... So we scientists, okay, we aim to be like 95% sure, okay? So 95% is right here, okay? If your chi-square falls within the range, so below this, it means that it supports your hypothesis. If it falls outside of it, it means it does not support it, okay? Make sense? So then here, this is 2.5%, so 97.5% confidence, 99, etc. Okay? The more you go this way, the more confident you are in your data. Okay? What else? What else? So we'll come back to if we're rejecting or accepting it. Okay? But just to review again, so we make a hypothesis that the genes are not linked. That's step number 1. Step number 2, we calculate the chi-square value, and we do that by using this formula here and this table. I suggest you do it this way so it's easier for you. And then we compare the chi-square to the threshold of 95%, so 0.05 for the p-value in this table. Okay? And if it's above, we reject our hypothesis. If it's below the number found here, we accept our hypothesis. Okay? Obviously, there are other explanations that can happen here. Sometimes, if we play me and you rock, paper, scissors, the chance that I get 10 times rock is very low, but I could just do like rock, right? So there's other explanations to this, right? It's not just chance or whatever. Yeah?
So are we rejecting the upper because the table we're using is the lower end table?
Sorry?
Because there's 2 chi-square tables. Are we taking like the lower end tables or
2 chi-square tables?
maybe not? There's the lower end and the upper.
This is the first time I hear this. There's a negative.
No, it's like the other ones are closer to 0. This one.
These ones here? I don't... No, seriously.
Well, maybe they fuse them together. Yeah, okay, never mind. Usually just this table is like split into 2.
Okay.
It goes from 9 to small.
You have to show me this table that you're talking about, okay? First time I hear this, okay? Yep.
Why don't we take 12.838?
Why don't we take 12.838? It depends on what's your hypothesis, what's your confidence that you choose, okay? If you're looking for 95% confident or more, okay? So this is something that scientists do. For example, if I'm using a technique that is very precise, I'm okay with 1% confidence. If I'm using a technique that's a little bit not perfect, I'm okay with, more margin of error, let's say, okay? And this I know it seems like a little bit arbitrary, but when you do research, you have to adapt the stringency of the method that you're using to the technique you're doing, okay? So in the exam, what concerns you is 95%, okay? That's the general scientific accepted threshold. Yeah?
You want us to round
Round
down?
down?
Yeah, like here it's between 11.3 and 12.8.
No, so you would look, so if I'm asking you for a 95% confidence, you will look at this column here, this column here. You find this number right here, 7.815. If your chi-square is above, you reject the hypothesis. If it's below, you accept it.
So you don't want the specific p-value?
No. So, yep.
Yes.
Okay, good. Does this make sense, guys? Yep. That's it. No, we reject the hypothesis that they are not linked, so they are linked, which is the hypothesis. Yeah, exactly. They are linked. Excellent. So our initial hypothesis was that the genes are not linked. We found the chi-square being above the threshold for 95%. And so we reject, in statistical language, we reject the null hypothesis. In English, the genes are linked, okay? Yeah. Yeah.
Starting from the hypothesis, then how did we translate that to numbers? What would have been different if we switched the hypothesis?
If we switch the hypothesis,
How does the
the expected values would change. Because the expected values for them to be linked is 300, right? And then if it's something else, it would be a different expected value, okay? But there will never be any other than this scenario, okay? This is the only scenario, follow it, that's it, okay? But yeah, based on your hypothesis, then you're expecting something, right? And that's your expected value, and then you compare it to the observed, okay? If we said that they are linked, then the expected would be different here. Yep.
So in that case, are we 99% sure that of the null hypothesis or the opposite?
Whoa. Say that again.
In that case, after the assumption of having the null hypothesis,
Yeah.
99% is with the null hypothesis or the opposite way?
So if so now we're rejecting it, right? So we're 95% sure that it's not that. Does that make sense?
Yeah.
Kind of. Yeah, just have to think in opposites. Opposites. Okay. Yeah, so it's all written here. If the chi-square value is larger than the 0.05 threshold, we reject the null hypothesis. Okay. There's a technique that you learn. Yeah. Yeah. So there's something that you learn in organic chemistry, mass spectrometry. It's super precise, okay? It goes to like 0.00001. So there, I don't mind having a lower threshold because I know that the data is super precise, you understand? But sometimes when the data is really bad, like the technique is not precise enough, then you have to be more stringent. And I think earlier I said the opposite of this, okay? But yeah, just 1 second. Yeah, go ahead. You're talking exam or real life? Real life, no. Real life, you will make your own hypothesis if you can, because we can make the hypothesis theoretically that the genes are linked, okay? And for them to be linked, you're going to end up going into probabilities, and the equation will become like this, because you'll say, I assume that the parental will be 80% of the progeny, so 80x, and then right? So to make it easier for the exam and the students at this level, we just assume they're not linked, because we have the expectations, right? We can measure them without doing too much algebra, right? Yep. That was the question. Okay, cool. In science, in research, it's a bit different. Okay. You have to figure out your expected, and so you go into the math of it and then try different things until you figure it out. Okay, let's test you out. So let's say we calculate a chi-square of 5.37 in an experiment with 4 categories. Do we reject the hypothesis or accept it? Yep. We accept it. Let's see. So how did you do that? So 3 is the categories, right? So you do 4 minus 1. So that's your degree of freedom. And then? Yeah. We accept it. We accept the hypothesis because the 5.37 is below the threshold. Oh. Right? Good. Easy stuff, I hope. Okay? Yeah.
So to assure is always
Yeah, for our purposes, always 95. Yeah. Yep. So you guys are good? Okay. One thing I forgot to cover at the beginning of last lecture is the concept of genetic maps versus physical maps, okay? Which is very simple. Genetic map is what we've been doing. It happens by recombination and all that. Physical map is the base pairs, so the nucleotide sequence, okay? So when I showed you on the browser there, USC, whatever, that's a recombination map, okay? Because you see the genes, you're not looking at the sequence, okay? Make sense? And they're not, obviously one is more precise than the other. The physical map is much more precise because it has the nucleotides, and so you know exactly what's in there, okay? Cool. A little bit more questions. We're almost done. I think I'm never going to do the lecture 4 or 3 pedigrees there, okay? So what did I want to say? So if you have a map like this, okay, with 4 genes, and AB has a high rate of recombination, CD has a low rate of recombination, which region will have the largest genetic distance? Anybody? Yep. Why? Exactly. So A and B will have the longest distance because there's more recombination happening due to more space, more chance of a chiasmata to happen. Okay? Cool. A little bit of interesting stuff. Okay? So you've heard of 23andMe, right? And Ancestry and all that. So they use these techniques to figure out, you know, the genotype of people and then figure out how linked to, let's say, I don't know. For me, they gave me 4% Nigerian and some stuff like that. But it makes sense, right? North Africa, so it's close to there. So for sure, there's some roots there, right? And they use this kind of stuff, right? Also, for example, the people that are coming, so Dylan, who's coming to talk to you, he's using a lot of this stuff to take a gene from one species, put it into another, and then use its properties. For example, they use a microalgae, which they modify genetically by just changing a gene in there. And the gene is just more efficient at converting bad fat into good fat. And so now he has algae that are rich in omega-6. How is that interesting? It's very expensive, okay? So they sell it for fish feed and all that, okay? So the omega-6 is probably produced that you buy omega-6, omega-3. So people believe that one is better than the other, by the way, but it's not that. It's the balance of both, okay? Keep that in mind, because I see people just buying, you know, supplements left and right. It's more complex than that. Finding family relatives. So let's say you lost someone and you're trying to figure out if he's part of your family or not, or, you know, kids, if they're your own kids. Hopefully that will never happen, but, you know, sometimes it happens. Then you also use these techniques because you compare the genes, right? You take them out and then you compare them to other people, right? And these centimorgans are not that useless. They can be used to figure out if you have cousins, if people are your cousins or not, okay? So genetic counselors like to create words here that are useless. Once removed, okay? What this means is that first cousin once removed, it means the son of your cousin, okay? Or the daughter of your cousin. It's just instead of writing daughter, they once removed, okay? Stupid stuff, but anyways. So if there's 450 centimorgan similarity between you and that person, then it's probably the child of your cousin. And then you can draw the whole map. Yeah? DNA. Any length, anywhere in the DNA. So you could take, as long as you have more close to 450 centimorgans of similarity in the DNA, then you're officially it's your your cousin's child, okay? And then if you're looking for a second cousin, so let's say your dad has his own cousin, then the kid, that's your second cousin, it's about 250 centimorgan. And there's numbers for each part of the tree, okay? Make sense? And this is like conserved. It's not, usually it's 250, 450, and then there's other numbers, okay? Good. Testing you guys. More questions. So let's say we have these two configurations, okay? The chromosomes had a crossover here, they're leaving, and here there was no crossover, okay? If A and B are 10 centimorgans apart. Which chromosomal configuration will be more frequent? Left side or right side?
Right side,
Yeah, why?
because 10 centimorgans is 10% probability of a crossover.
That's it. So 10 centimorgan means that it's 10% probability that a crossover will happen between those two genes. So the side here, the left side here, is less frequent than just staying like this, okay? Because the parental are these ones here at the top. C'est bon? Good. Question for you guys. Can you find the gene sequence if I give you these numbers like this? Try to do it. Find the biggest, find the smallest, and then start from there. 20 and 28, 28 and 20 and 8, they seem alike, right? So start from there. Yep. A, B, C, D. So distance between A and D, 25. B and D, no. Because the distance between A and D is 25, right? And so, and B and D is 33, so this is impossible. B and D is bigger than A and D. Yep. B, A, C, D. Yeah. B, A, D, C. Let's see. So distance between B and C is 20. So that's not possible because distance between A and C is 28. The real answer is D, okay? Why? We did all of them. Why is it this one, okay? So B and D is 33. C and B is 8, okay? C and D, we don't have anything here, so that works. A and B, where is A and B? 8, and then A and D, 25, and B and D, 33. So 8 plus 25 gives 33, so this works here, right? Make sense, guys? Yeah. And then C and D, we don't have the distance, so that one works out. Yep.
A and B, does it matter? They are both ABCD.
What? Sorry?
A is ABCD, B is DCBA.
Ye- No, it doesn't. No, it doesn't. Yeah, it's the same thing. Yeah, just flipped. This is called laziness when you're making a question. Still works though. Still works. Anyone has a question about this question? Yep. C, D. D. Yeah. Okay, lecture 4. Why am I covering lecture 4? Maybe because it's in your tutorial quiz next week. Okay, so focus. Okay, so there's only a few slides. I think 1, 2, 3, 4, 5. Okay, so it's just a question about the pedigrees. Okay, you need to be able to figure out the type of pedigree it is. Okay, so if I give you a pedigree without writing all this stuff here, and then first of all, interesting fact. Okay, albinos used to be believed that they were. Albinism was believed to be caused by inbreeding. Okay, so family breeding, just some racism BS. Okay, scientists showed that was wrong. Okay, that's why I put this here. Now back to this stuff here. So if I give you a pedigree without telling you all these things here, you should be maybe just give you these guys here. You should be probably able to figure out if it's homozygous. If it's recessive or dominant, if it's autosomal, or if it's sex-linked or organelle-linked, okay? So this is why I'm focusing on this. So if you look at this one properly, usually when the pedigree is empty like this, it's because it's recessive, okay? If you see one, if you see one in each generation, most probably it's because it's dominant. Okay? For example, color blindness usually skips one generation. Okay? And so if a male has it, usually his mom is a carrier, and the mom, her dad has it because he gave that X chromosome. Usually, okay? So male has it, mom carrier, the dad of the mom has it. Okay? The grandfather of the person. Can I ask you, if it's not too personal, does your grandfather have it? Yeah. Not sure? Okay. Yeah. So there's something called genetic counseling. You can actually it's a specialty that you can study probably at McGill. They do a lot of this kind of stuff here. Predicting, well, not predicting, but seeing if you're going to have problems with the kids or not, and whatever. Okay. There's genetic diseases also that is important. If, for example, a family has breast cancer, then you can figure out who's going to get what, right? And it's pretty precise. Okay. X-linked, that's a bit different. So X-linked, if it's there's two types also. If it's recessive or dominant. If it's recessive and the male gets the X chromosome that's messed up, he will have the disease. Okay. Same thing for so for female though, that's a bit different, because then it becomes like an autosomal gene. It follows a 3-to-1 ratio. If it's recessive, X-linked recessive. If it's dominant, same thing. You will see that it follows this kind of pattern. If it's X-linked, that means each generation will have it. Okay? Daughter or son, doesn't matter. Okay? See what I mean? Because it's dominant, right? So even if you have one good copy and one bad, that's it. Yeah? Well, not necessarily at the bottom, but I will give you something that will give you a clue in there. And then you build from there. Okay? My favorite one is the mitochondrial one, okay? The organelle transmission. So this one is pretty simple, okay? If the mom has it, everyone has it. It's that simple, okay? You can see all the siblings, the kids have it. And that's it, guys. So one mistake I made when I presented this, I was talking about myopathy. What I meant is not eye problems, it's muscle problems, okay? I was watching the lecture and I was like, what the hell are you saying, okay? So don't mix that up. Myopathy means muscle problems, okay? I'm like recovering from previous lectures. Cool, that's it, guys. Thank you for listening. See you later.